In this last video on respiratory anatomy physiology, we'll look at some of the details of respiratory physiology, including the process of respiration, ventilation, talk about something called lung compliance, airway resistance, lung volumes, look at the transport of oxygen and carbon dioxide in the blood, and then talk about respiratory acidosis and alkalosis. So there are 5 basic steps to respiration. So the first is the ventilation, which would be the exchange of air between the atmosphere and the alveoli by what's called bulk flow, which is just the flow of the gases. Typical breath is about 500 milliliters or half a liter of air. And the frequency at quiet breathing is about 12 to 16 times per minute. And that's about 6 to 8 liters of air per minute. The gas exchange is at the level of the alveoli, oxygen, CO2. Really, O2 is diffused between the alveolar wall, through the capillary wall, and into the hemoglobin in the red cells. About 500 milliliters of oxygen is inhaled, but only about 350 milliliters of that crosses into the alveolar capillaries with each breath. And about 150 milliliters remains in what's called the anatomic dead space of the lungs. So interestingly, there's always a little volume of air in the lungs. Even if you try to maximally exhale, some air remains in the lungs, and that's called the anatomical dead space. About 200 milliliters of carbon dioxide crosses into the alveolar spaces and is exhaled. The transport of oxygen is via hemoglobin, and carbon dioxide is transported to the lungs. Primarily dissolved is carbonic acid in the blood. So we'll look at that pathway here. That will be the exchange of those two gases. And then at the cell level, oxygen is going to leave, dissociate from the hemoglobin. So hemoglobin has an interesting feature. We're under low oxygen tension. It changes conformation, and that allows the oxygen to dislodge. And the oxygen then diffuses into the tissues, and it's able to get across cell membranes. It gets right into the mitochondria where it's needed for aerobic metabolism. And then the carbon dioxide moves the other way. And then at the cell level, we have the mitochondria, and that's what uses the oxygen and produces carbon dioxide as a byproduct of aerobic metabolism. And then the CO2 goes in the other direction. So that's the whole process of respiration. Again, the first part here, ventilation and gas exchange, really is part of pulmonary respiration. And then cellular respiration is the last part here at the cell level. So let's break this down into individual pieces. Let's look at ventilation first. So that is the exchange of air between the alveoli and the atmosphere through bulk flow. The air really moves from a region of high air pressure to a region of low pressure. And the most significant factor that changes the alveolar air pressure and thus drives the air into the lungs is the change in lung dimension. And we've already talked about how the lung is lined by the 2 layers of the pleura. The parietal pleura is attached to the inside of the thoracic cavity, thoracic wall, and the visceral is attached to the lungs, and there's that little intrapleural space with a slightly negative pressure in between. So if you increase the dimensions of the thoracic cavity, that will pull basically the parietal pleura, and that will pull the visceral pleura, and then that opens the lung. And that's going to be the primary change that allows for bulk flow. So in the resting state, between breaths, the lungs are prevented from collapsing via the slight negative pressure in that intrapleural space. So that holds the visceral pleura pretty much tightly kind of towards the parietal pleura. So that holds the lungs open. And it's that negative intrapleural space. Of course, if that is damaged, and like in the case of pneumothorax, that could collapse the lungs. In the resting state, there's no movement of air in or out of the lungs. So the alveolar pressure is the same as atmospheric pressure. On inspiration, the outside air moves into the alveolar spaces, and that's due to decreased alveolar airspace pressure. And that's to do with, I've already mentioned before, how the diaphragm contracts, and as it contracts, it creates a larger thoracic cavity. And that opens up the chest, and that will draw air into the lungs. And so diaphragm at rest is the primary respiratory muscle. The inspiratory intercostal muscles also assist that, but that's usually not under rest, that's under more active breathing. And again, it's the phrenic nerve which fires, which is going to cause the diaphragm to contract. The lungs expand, the alveoli enlarge, and the alveolar pressure drops. So a good little demonstration of that is to have, for example, if you had a glass bottle and you have a stopper with a little tube and it branches into two tubes when they have balloons attached at each end, and then a rubber diaphragm at the end. If you pull down on the diaphragm, that will actually decrease the pressure inside the bottle and the balloons will inflate and that'll pull air in from outside. And that's exactly how the lungs work. And so that's inspiration, expiration, the diaphragm relaxes, the balloons or the lungs collapse, and the air leaves the lungs. So this is completely an active process. It does require energy, and that's the energy to contract the diaphragm. Expiration is going to be the relaxation of the diaphragm and the intercostal muscles, which results in a recoil of the chest wall and the lungs to their original size. Increases the alveolar pressure, and that drives the air out of the lungs. So expiration is a completely passive process, requires no input of energy. During exercise, other muscles can be recruited to assist with this process. So again, the diaphragm is active in quiet breathing, but in inspiration, we can have some accessory muscles come in. So your sternocleidomastoids, they can elevate the sternum. The scalenes, which are in the upper ribs, can elevate the upper ribs. The pectoralis minor also can participate there. And then we have some principal muscles. Again, the diaphragm is going to be the main muscle, and our external intercostals will also be active to some degree at rest with inspiration. With expiration, with quiet breathing, it's just the elastic recoil of the diaphragm. No other muscles are recruited. But with active breathing, like during exercises, the internal intercostals can pull down on the ribs, and your abdominal muscles can also pull down on the rib cage. And so that creates a smaller interthoracic cavity, and that drives air out of the lungs. So that's the difference between more active and breathing at rest. So that's the general process of inspiration and expiration. Again, slightly different muscles are recruited in the more active inspiration and expiration. Now, how easily air gets in and out of the lungs depends on two factors. One is what's called lung compliance. How compliant, how stretchy are the lungs? And that's how easy it is for the lungs to expand based on the change in the transpulmonary pressures we just talked about. This is really the more compliant the lung is, the less stiff it is, and the more stiff the lung is, the less compliant. So the less compliant, the more energy is going to be needed to actually get the lungs to expand. And the determinants of that will primarily be elasticity of the lung tissue. So there's a lot of elastin in the connective tissue in the lungs, and especially in the alveolar walls, and that improves compliance. And then the surfactant. Remember, surfactant decreases that surface tension, and that will make the lungs more compliant as well. So if you have a decrease in elasticity and or surfactant, less compliance, more difficult it's going to be to inspire in the lungs. And the surfactant is really an issue more with prematurity or anything that damages the surfactant. It's really like a detergent. It has phospholipids and proteins. And so we need that to keep the lungs, the airways open. Not the airways, but the alveoli. The elasticity can become damaged in chronic inflammatory conditions like an emphysema, where we have a collapse of the elastin around the alveoli. And we'll look at that in detail, but that typically results from a chronic inflammatory process from smoking, cigarette smoking, but it can actually result from a genetic condition called alpha-1 antitrypsin deficiency, and we'll talk about that later. Respiratory distress of the newborn is really when premature babies are born without that adequate surfactant production, and that results in really decreased lung compliance, inability to breathe, lung collapse, even death. And this is actually the leading cause of death in premature infants, is lack of lung compliance. The other issue when we're talking about the ease at which air can enter the lungs is airway resistance, and that is how open are the conducting zone airways, so the bronchi, primary, secondary, tertiary, all the way down to the bronchioles. Usually the airway resistance is very low, but if there is constriction of the airways, bronchoconstriction, that will increase the resistance. And so we see that in conditions like asthma, COPD, for example. And the determinants really of that are transpulmonary pressure. So the opening up of the thoracic cavity when the diaphragm contracts, for example, or those external intercostal muscles pull up on the ribs, they help to increase the radius of the airways, and so that decreases their resistance. Lateral traction has to do with forces of elastic connective tissue that opens them up, also with the breathing process. But the most important factors here are probably neuroendocrine, and that would be the factors that influence smooth muscle, especially on the bronchioles. And so I already talked about how the sympathetic nervous system, epinephrine and the norepinephrine really relax. They cause bronchodilation. And then leukotrienes, which are inflammatory mediators, they tend to cause bronchoconstriction. And so again, if we look at asthma, we see there's an increase in airway resistance. So we give the beta-2 agonists that we already talked about. They would mimic epinephrine. They bronchodilate. And then the leukotrienes, actually there are medications, leukotriene inhibitors, that can block the leukotrienes. And that can help with the airway resistance as well. Now, there are a couple of important physiologic measures which can help us kind of measure the volume of air going in and out of the lungs. And this is going to have a clinical application because it's going to be very helpful for differentiating clinically different types of respiratory disorders. There is a machine called a spirometer that can allow us to assess these pulmonary capacities and volumes. So a couple of definitions here. First of all, the average capacity in an adult male is about 6 liters of air in the lungs total, a little bit less in females. The average respiratory rate is between 30 and 60 breaths per minute at birth, then it decreases to 12 to 20 breaths per minute in adults. The total lung capacity is basically the theoretical air capacity of the lung tissue. So how much can you maximally inspire, how much can you maximally expire, and then that physiologic dead space. So that dead space is called the residual volume, and this diagram to the right here can help us with that. So that's residual volume, and then the total lung capacity, TLC, is basically, if you look at this picture, this little wavy line, this is at rest. So with every inspiration, we take in some air, expiration, we go out. So this is our resting capacity, and that is known as the tidal volume. So that's the amount of air that is inhaled, exhaled under normal respiratory conditions. So that's about 500 milliliters again, depending on the body size. Then if we maximally inhale, like how much can you inhale until you can't inhale anymore, and then how much can you actually maximally exhale as much as you can, that is the vital capacity. So vital capacity is on top of your tidal volume, how much you can maximally inhale versus how much you can maximally exhale. And then we have things like what's called the expiratory reserve volume. That's basically under the tidal volume with your maximal expiration, how much air is there, and then how much can you maximally inhale here. That's the inspiratory capacity and the inspiratory reserve volume, etc. So lots of different definitions there. So these measures are important because we can actually measure them with a machine, again, the spirometer, that allows us to breathe, take deep breaths in, do a maximal exhalation, maximal inhalation, and it can measure how quickly it takes us to do that. So we get different measures, and we'll look at these later. For example, there's something called forced expiratory volume. You breathe out maximally as fast as you can, all the air in your lungs, and it measures how much you can breathe out in 1 second. And that's called the FEV1, forced expiratory volume in 1 second. And then we can look at ratios between that and the total vital capacity, and that can give us some important measures of lung compliance, and so forth. So it really helps us measure the difference between what's called obstructive lung diseases, and those usually are from obstruction in the airways, from decreased airway resistance, versus restrictive lung diseases, which have to do with problems of the lung compliance. So a lot of fibrotic lung conditions, the lungs are less compliant than in obstructive diseases. So we'll talk about those in the assessment of lung disorder section. So once the air has entered the body, has diffused across the alveolar wall, it's going to diffuse into red blood cells, and it's going to attach itself to hemoglobin, and that's what's going to carry it to the different tissues. There's a little bit of oxygen that is dissolved directly in the plasma and in the water in the blood cells, and that is about 1% of the total dissolved oxygen, so that's very small. So most of it is reversibly combined with hemoglobin. Now, hemoglobin is an interesting molecule. It's packed into red cells, and it's made of 4 basic subunits. So it's made of protein, and it's made of 2 protein chains. These are called globins. So there's 2 beta globins and 2 alpha globins. And then in the middle of each globin here, there's a molecule called heme. So there are 4 heme molecules in each hemoglobin molecule. And at the middle of heme, there's iron. And it's the iron that attracts the oxygen. So the oxygen is going to nestle in there. So each hemoglobin molecule can carry 4 molecular oxygens. That's 4, O2 is molecular, that's what's in the atmosphere, molecular oxygen. Now, interestingly, this same molecule is found in plants and chlorophyll, but instead of iron, the central molecule is magnesium. So kind of a curious thing there. And if you look at adults, our primary form of hemoglobin is called hemoglobin A. In the fetus, there's slightly different hemoglobin. The globins are different, and that's hemoglobin F. And there are several variants. For example, in sickle cell anemia, we have hemoglobin S, and that's to do with a mutation in the globin chain that causes it to actually buckle, change conformation in low oxygen environments, and that causes sickling in the red cells. We'll talk about that in the hematology section. So there's different variants of hemoglobin. For adults, hemoglobin A is most predominant. In the fetus, hemoglobin F actually has a higher affinity for oxygen, so it can function better in a low oxygen environment. But that's going to be quickly replaced in the first couple months of life with hemoglobin A. Again, each heme group has a molecule of iron in the +2 state, and that's going to bind the oxygen. Now, hemoglobin without oxygen is deoxyhemoglobin, and with oxygen is oxyhemoglobin. There's a measure of the percent oxygen saturation of hemoglobin, and that is how much oxygen is actually bound to hemoglobin, forming oxyhemoglobin, and that's usually going to be determined by the partial pressure of oxygen in the blood. Under resting conditions, it's usually about 75%. So 75% is the saturation of hemoglobin. And so that actually, even when the blood leaves the tissue capillaries, it still contains 75% oxygenated hemoglobin. And so this gives a lot of extra capacity for that hemoglobin to be needed for like exercise and things like that. In areas of low oxygen pressure tissues, the oxygen dissociates. There's something called the Bohr effect, where basically the oxygen is able to dissociate from the hemoglobin and it diffuses into the tissues. The presence of local tissue acidity or increased temperature will actually shift that Bohr curve to make it more likely to dissociate. So it'll accelerate dissociation. So when you're exercising, you create acidity in the tissues. There's a need for oxygenation. More oxygen will dissociate from the hemoglobin. Now, unfortunately, carbon monoxide also binds to that same site with much more affinity than oxygen. In fact, 210 times stronger affinity than oxygen. And so, unfortunately, it would then block the ability of oxygen to bind to hemoglobin, and that can create hypoxia. Unfortunately, blood that has been saturated with carbon dioxide Normally when when the hemoglobin is deoxygenated, it turns bluish. It looks blue in visible light, whereas oxygenated hemoglobin looks more red. That's why your arteries look more reddish, your veins look more bluish. Carboxyhemoglobin, which is basically where you get carbon monoxide binding to hemoglobin, actually looks very red, cherry red, pink. So it actually confuses the pulse ox monitors, the things that measure oxygen saturation, those little clip-on monitors. They're actually shining light to the skin. They're measuring the absorption spectra through your skin, and they can actually determine the amount of oxygen-saturated hemoglobin from that. And that's where we get the oxygen saturation. But basically with carboxyhemoglobin, that would confuse that. So we can't use those monitors in those cases. Nitric oxide can also bind to hemoglobin. And what's interesting about nitric oxide, NO, it's kind of the gas that allows for vasodilation. And so as it goes around, the hemoglobin goes around inside the red cells, it can actually release nitric oxide, and that can cause vasodilation, allowing blood to flow more easily. There's conditions when that nitric oxide just doesn't work well. Endothelium normally secretes that, but if it doesn't secrete it adequately, we'll have more constriction of capillaries and small arteries called arterioles. And that is part of the picture we call in Chinese medicine, for instance, blood stasis. So that'd be a state of decreased nitric oxide. And that happens often from a state of increased systemic inflammation. So that was oxygen, but how does carbon dioxide get from the tissues back to the lungs? Well, there's really three ways this happens. In a person at rest, the metabolism just generates about 200 milliliters of carbon dioxide per minute. So all that has to get back to the lungs. And there's three ways it can do that. One is it can be directly dissolved into the plasma. So carbon dioxide is about 20 times more soluble in water than oxygen. So you've probably seen carbonated water. That's just water with carbon dioxide dissolved into it. That accounts for about 7% of all the dissolved CO2 traveling back to the lungs this way. Another 23% or so is basically bound to hemoglobin. And that forms carbaminohemoglobin. And this is another way the hemoglobin can directly then take that to the lungs, deliver it. It dissociates from the hemoglobin, and then we breathe it out. So that's a second way. But the majority of carbon dioxide is actually dissolved in the blood as bicarbonate. And I'll talk about that here next, but that's going to be the major transport mechanism of carbon dioxide. The total blood carbon dioxide is the sum of carbon dioxide from all these forms. And this can actually be measured as part of routine blood chemistry testing. We can actually measure the total amount of CO2 in the blood. The bicarbonate buffer system is what allows us to essentially allows carbon dioxide to dissolve as bicarbonate. And again, that can carry it back to the lungs. So basically, this requires the enzyme called carbonic anhydrase. And that's found inside a red blood cell. So if we look at tissues, they make CO2 that gets into circulation. In the red cells, carbonic anhydrase combines carbon dioxide with water, and that makes bicarbonate or carbonic acid, H2CO3. And then H2CO3 dissociates into bicarbonate anion, HCO3 with a negative sign, and a proton. And that, of course, is acidic. Most of the protons, fortunately, combine with hemoglobin, so that neutralizes the acidity. Some of it does, though, unfortunately, get out into the plasma. And so as you carry more carbon dioxide, you actually make more hydrogen ions, and that makes the blood more acidic. So very important factor here is that the more carbon dioxide there is in the blood, the more acidic your blood becomes. And so the lungs, as we'll see, have a very important role by ventilating carbon dioxide, preventing blood acidity. We'll come back to that here in just a moment. Now, as the bicarbonate is made, the bicarbonate ion, it's going to come out of the red cells, go into circulation, and then chloride ions will come from circulation into the red cells, and that's called the chloride shift. Now that's going to go to the lungs, and in the lungs exactly the opposite factor process happens. So HCO3 goes back inside the red cells. It's combined with the hydrogen ion from hemoglobin to become the bicarbonate, carbonic acid. And then it's going to dissociate via carbonic anhydrase back into CO2 and water. And the CO2 now will diffuse out of the red cell into the alveolus and be exhaled. And chloride will have shifted back from the red cell back to circulation. So complicated process, but that is how the majority of CO2 is actually carried around the blood. Now I mentioned that most of those hydrogen ions that are generated in that above process are buffered by the deoxyhemoglobin, so hemoglobin that has been dissociated from oxygen. And this prevents the venous blood from becoming too acidic. However, there is a little bit that does leak out, and that creates a little bit of acidity, but basically arterial blood is normally of a pH that's basic, so 7.40 to 7.35. So slightly different in the arterial versus venous blood. We usually say between 7.35 and 7.45 is our normal blood pH. Now, what's important about that is really we have to maintain that very narrow range for all your cells to function. Anything above or below that is going to cause significant problems. In fact, you're probably going to be in the ER if it's going to go above or below that. So there have to be these buffer systems that come into play. In the lungs, one of the buffer systems, of course, is to ventilate out the carbon dioxide. And so that will, the CO2 will combine with those hydrogen ions, and that'll prevent the hydrogen ions from going into solution, and that will take the acidity out of the blood. However, in different lung conditions, this can be a problem. I should say there's really two organs here that are going to balance those hydrogen ions. So the lung is one, but the other is your kidney. So the kidney system, we can say the lungs are working minute to minute short term to regulate acid-base balance in the lung via that bicarbonate buffer system. The kidneys are going to work more long term to get rid of acid waste. So there's a lot of other acids your tissues make, and the kidneys are really responsible for that. So lung conditions, lung diseases, and kidney diseases can result in problems of acid-base balance in the blood. So it's possible to develop 4 different conditions. Respiratory acidosis or respiratory alkalosis, that's where the blood becomes too acidic or alkaline, or metabolic acidosis or metabolic alkalosis. And we're going to talk about the metabolic ones in the kidney section later. But in terms of respiratory acidosis and alkalosis, in respiratory acidosis, too much carbon dioxide stays in the blood, and we get saturation of the CO2 in the blood, and that's called hypercapnia. And we often see a bounding pulse with warm, moist extremities, drowsiness, dizziness, disorientation, a lot of muscle weakness, hyperreflexia. In general, this is from a decreased alveolar ventilation. So either a decreased breathing rate, problems in the brainstem with that respiratory center we spoke about earlier, or chronic obstructive pulmonary disease where we don't have proper ventilation in the lungs. So that can create respiratory acidosis. That's going to put extra strain on the kidneys, but the kidneys often can't keep up with a lot of that acid waste. Alkalosis is when we have too much CO2 that leaves the blood. And that can present with palpitations, tetany, convulsions. We get too jumpy in a way. And interestingly, this is usually due to increased alveolar ventilation, and especially with anxiety or stress where people hyperventilate. They blow off too much CO2. And this is why we sometimes have people that are in a panic attack breathe into a paper bag so that they re-inhale that CO2 so it can keep their blood from becoming too alkaline. Different pulmonary disorders, altitude changes, salicylate. Salicylates are the precursor to aspirin. Poisoning from that, hyperventilation again, and then liver disease can all cause respiratory alkalosis. So that's kind of the two polarities there. Again, we'll look at metabolic acidosis and alkalosis with the kidney section, but this is the role of the lungs to maintain that. Now, this obviously has a really important effect on consciousness, and we can say that there's a more subtle regulation constantly of carbon dioxide in the blood by the lungs, and that's going to affect your thinking. So that's kind of bringing in this interesting idea that the lungs have a connection with our thinking processes. In the traditional medicines, we say people are very intellectual, very linear in their thinking, and so forth, have an overdeveloped lung activity, and we might liken the relationship there. Another potential connection with the brain and thinking and the psychic aspects would be the breathing rhythm sets the rhythm of the cerebral spinal fluid. And we actually know that people who breathe through their nose, inspire through their nose, actually have better memory and can memorize things better than people that breathe through their mouths. So that was actually a study that was done. And so that shows another connection with the breathing process and, in this case, mentation. So don't forget respiratory acidosis, alkalosis. We'll be looking at those with a number of different disease states. So we've already talked a bit about the control of respiration. This, of course, depends on the breathing centers, the dorsal and ventral respiratory group in the medulla, and the pontine respiratory group in the pons, in the brainstem, both of these, that are going to really trigger the phrenic nerve and the nerves that go to your accessory muscles for breathing. So this is the respiratory rhythm generator, and it really mediates normal tidal breathing rhythms. Now that rhythm can be increased or decreased based on additional inputs to the rhythm generator, and that would be either through peripheral chemoreceptors, and that would be in your carotid arteries or your aorta. There are sensors there, and they're found actually the bifurcation of the internal and external carotid arteries and the neck and inside the aortic arch. Close to they're close to the arterial baroreceptors, and they're in intimate contact with the blood, and they're monitoring the partial pressure of oxygen and hydrogen ion, so blood acidity levels. And basically, when the oxygen levels are too low, or more particularly when the hydrogen ions are too high, usually meaning there's too much carbon dioxide, that's going to trigger the peripheral chemoreceptors, and that will trigger breathing. The chronic arterial hypoxia, so chronic low oxygen levels, can actually induce those carotid bodies to become hyperplastic, and they start to fire incorrectly. So we'll talk about that with a couple of chronic disease states. Central chemoreceptors are located in the medulla, and they're most sensitive to the hydrogen ion content, and again, carbon dioxide levels, especially in the extracellular fluid of the brain, and that would be tied in with the cerebral spinal fluid. So if the hydrogen ion levels rise too quickly, that triggers inspiration. So that would be another signal to send out, to activate these centers, to send out more impulses to start breathing. These medullary inspiratory neurons are very sensitive to different drugs like barbiturates and morphine, and this is why overdoses of these medications can result in respiratory failure. Those centers stop firing, and people die via respiratory depression. So the primary metabolite influencing respiration is blood hydrogen ion concentration, and that's directly related to the carbon dioxide levels. Thus, carbon dioxide, not oxygen, is the primary driver of respiration. So it's interesting to step back and look at respiration more from a phenomenological view. We can think that the process of inspiration is really the process that awakens consciousness. It really increases your animal waking forces. We can say from a subtle body perspective that it's an incarnation of the Hun and the astral body. This is what really lights up consciousness. We get an increase in oxygenation, arterial blood flow, increase of the mitochondrial activity, which makes more ATP. And this is connected with warmth as well. When you make more ATP, you also make more warmth in mitochondria. Heat is a byproduct of that process, especially in muscle. That whole process requires a lot of phosphorus. So ATP, of course, is adenosine triphosphate. That's phosphate. We have phosphorus involved in a lot of the other processes that are involved in energy making. So we can think about this as phosphorus is kind of, we might call this an incarnation element. It helps us to actually ground ourselves in the body. So inspiration, it's as if our soul spiritual element is coming in, connecting with the physical tissues. It's really a reverse of what's happening in plants. Plants are taking light and they're storing it in the form really of sugars. And we are actually doing the opposite. We're releasing it. And this process depends on the phosphorus, as I just mentioned, but also iron. So we can think of iron in the hemoglobin as part of that as well. So think of iron as an incarnation metal. And when you think about people with iron deficiency anemia, they're usually very sleepy, tired, pale. It's like they're not well incarnated with light and warmth. You give them some iron and now they perk up. Their cheeks get red, they start to, their nail beds start to perfuse better, etc. Same with phosphorus. In fact, iron phosphate, ferrum phos, is an old homeopathic cell salt that's often used for people that have that anemic, kind of low inner light, low inner warmth kind of picture. So some have actually referred to iron really as the primary respiratory metal. It's really what's helping the breathing. Now, interestingly, in the mitochondria, in the what are called the cytochromes, which help the electron transport to make ATP, they are dependent on iron. So there's another role for iron in that energy ATP making process. In the case of plants, that iron is replaced with magnesium, and that's more the green life activity, more sleep consciousness kind of thing. That's sort of the opposite effect here. In the head, we also have a lot of silicic acid forces, and I can talk about that later, but that basically allows for more crystallization, that salt process in the nerve sensory activity. And that's the basis for thinking. And again, that paves the way for our higher bodies. Almost like light shining through a quartz crystal, we can incarnate through the nerves and through that activity. The light itself can actually have a formative effect. So again, working through the neurons, we've talked about how the nerves and the neurons really have a form-giving effect on the tissues, and that's a type of inner light that actually helps to shape you. So there's a bit of a destructive element there as well. So inspiration is bringing in light, it's bringing in conscious awareness, it's waking us up. The opposite of that would be exhalation. I say that's more plant-like, carbon dioxide, the carbonic acid and whatnot. And that really is making you more, remember when you get hypercapnia, too much carbon dioxide, you become more sleepy. And the consciousness kind of goes away, but you become more metabolic in a sense. So in respiratory acidosis, too much metabolism actually enters the head and so forth. Now we need a little bit of carbon dioxide to be retained, and that actually, we can say on a very interesting, subtle sense, dulls the brain a little bit. It deadens the nerves. But that little bit of CO2, by deadening the nerves, actually allows for the liberation of consciousness. And so that's where there's an interesting alchemical relationship here with CO2, deadening the nerves, increasing consciousness. If we combine calcium with that carbonate, we get calcium carbonate, and then that becomes fixed into bone. So it's like consciousness leads all the way down to the bone level as well. When there's lack of deep inspirations, the metabolism usually takes control, it fills out. So we can think of a person who becomes more fleshy versus a person who's really breathing in deeply, maybe not consciously, but they're getting that oxygenation is more thin. So you can say the ectomorph more emphasizes that oxygenation process, inspiration, versus the endomorph retains most of that. And they are more clouded thinking, more that metabolic process that predominates. So the lungs again hold a balance here between these two polar opposite sets of forces. Finally, if you look at Chinese medicine, lung functions, we say the lung governs respiration, specifically the exhalation process. That's interesting to think that, really the inspiration is active with contraction of the diaphragm, expiration is passive. The inspiration really emphasizes the oxygen part, and that actually is regulated primarily by the kidneys. In fact, the kidneys put out a hormone to regulate the amount of red cells you have and the amount of hemoglobin you have. So really, kidneys are more related to the oxygenation. We can say the kidney system draws in the air and anchors it, versus the lungs are more involved in the exhalation and the gas exchange that way. Say it regulates the chi, and that's your basal body energy. And we can think of course, ATP production, the mitochondrial activity is part of that. But we can also link the thyroid there, and I mentioned we can maybe think about how the thyroid is a carrying of the lung activity into the tissues, into the mitochondria. So one aspect of lung chi we can think of as thyroid activity. It also purifies the chi. We can think of that as the CO2 balance and the acid-base balance. Rules the exterior. Again, we might think here about, number one, the opening and closing of pores has a lot to do with the autonomic nervous system activity. And the breathing rhythm together with your heart can actually help regulate your autonomics. So when people are stressed in sympathetic mode, one of the best techniques is to actually regulate the breathing. And that can reset you into a parasympathetic mode. And that can change the way the autonomics function in the skin and elsewhere. Thyroid also plays into that. Controls the voice. We can think of that via the larynx and the vocal cords. Regulates the nose, the sense of smell. We talked about how the lung system is connected, obviously, with the olfactory epithelium. And then we think of the upper source of water, so it distributes the fine vapor to the body. Again, thyroid functions tied in with that, and we see frequently with hypothyroidism, low thyroid, puffiness, edema. People get what's called myxedema. It's edema in the tissues, and they become very sponge-like. So we can think of the thyroid assisting the lung in that action. And houses the Po, and this is your basal biofield that maintains your life activities. And so we can maybe think of the lung as having a connection with bringing in the from the environment some of the energy that's needed to maintain that body. It's also, of course, the basis for fixed thinking. We talked about the carbon dioxide. You hold a little bit of that back and actually cause a deadening function on the brain, which actually increases your ability to think intellectually. So that's kind of paradoxical again, where consciousness and life activities are polarities. And by holding the CO2 back, we actually can encourage more the consciousness activities. Lung physiology, again, we can think of lung yang is really related to the arterial blood flow, the oxygenation, and several hormones assist this, so thyroid and androgens. DHEA, testosterone could be aspects of kidney chi. The lung chi we can think about as the lung autonomic activity, the sympathetics, which are bronchodilating and vasodilating, and the parasympathetics, which are more constrictive, and parasympathetics encourage digestion, but they really slow down breathing. And then the lung, blood, and yin, we can think of that as the mucosal surface we talked about, including the surfactant, the respiratory mucus, the immune paint, which is the mucus with IgA, and the lung microbiome could all be part of the lung yin. So when we're talking about lung pathologies, we often see a breakdown in one of these functions, and there are specific herbal therapies, acupuncture protocols, etc., which can target these physiologic functions. So that does it as our basic introduction. Covered most of the concepts we need to know as we now jump into respiratory pathology in the coming videos.